Hello there. Just imagine that we are in a situation where we are in the chemical plant factory where the wastewater are being produced in the factory, right? So the main question is how they're going to ensure that the wastewater are being managed properly. So one of the methods is called absorption. Absorption is a unit operation where all the active surface materials are being removed by the interface transfer to the surface of the absorbent particle. So our main objective is to know the best fit isotherm for the absorption of the absorbent, either by Freundlich isotherm or Langmuir isotherm. So in this experiment, we are going to find the best fit isotherm for the activated carbon which act as the absorbent by manipulating two initial variables, which are the concentration of the absorbate, which is the methylene blue, and the concentration of the absorbent, which is the activated carbon. So let us see how does it works.
Hi everyone, I'll be explaining on the procedure of experiment 8, which is activated carbon adsorption isotherm and kinetics. Firstly, we'll prepare 5 different weights of methylene blue powder, which is 10, 15, 30, 45 and 50 mg. After that, we'll prepare 5 beakers of 1 liter water. So after that, we'll pour all the methylene blue powder into the water to form methylene blue solution. After that, we'll prepare 5 the same weight of activated carbon, which is 150 mg. Before we pour the activated carbon into the methylene blue solution, it will be allowed to mix under the agitator speed of 200 rpm for 3 minutes to ensure that the methylene blue solution is evenly distributed. So next we will be pouring the activated carbon into the beakers to carry out our adsorption process. After the solution is mixed for 10 minutes, the agitators is stopped and the solution is pipetted out. And the absorbance index of the solution is measured by a UV spectrometer. Before we measure, water is used as a control variable for the measurement. After the absorbance index of the methylene blue solution is recorded, the agitator is started and the steps are repeated. In the first part of the experiment, we vary with the mass of the methylene blue, whereas in the part 2 of the experiment, we varied with the mass of the activated carbon. Activated carbon are prepared in 5 different weights, which are 100, 150, 200, 250 and 300 mg. And the concentration of solution of methylene blue remain constant at 30 mg per liter. And the following steps are repeated from the procedure of experiment 1.
For this experiment, different initial value of methylene blue concentrations towards the adsorption rate are also studied. From the finding of the experiment, the higher the initial concentrations of methylene blue solution, the higher the adsorption rate. How does this occur? Higher initial concentrations of methylene blue indicates a higher amount of particles of methylene blue in the solution. Thus, a large amount of particles can be adsorbed on the surface of the activated carbon, leading to a higher adsorption rate. However, the adsorption rate decreases after a certain period of time as the methylene blue concentration can become the limiting factor. For smaller concentration, since only a small amount of methylene blue particles presents in the solution, less amount of methylene blue left in the solution to be absorbed towards the end of the process, even though the activated carbon does not reach the maximum capacity of adsorption. Adsorption gives the equilibrium relationship between the amount of methylene blue particles adsorbed on the surface of activated carbon at constant temperature. For this set of experimental data, we obtained 2 plot, which is the Freundlich isotherms and Langmuir isotherms. From the graph shown, both of the plot describes the system well with a regression value close to 1. Which adsorption isotherms fit the experimental data? For this purpose, we will look closely at the value of the regression model. For the Freundlich isotherm, the regression model value is 0.9985, while for the Langmuir isotherm, the regression value is 0.9995. Since the value of regression value of the Langmuir isotherm is closer to 1, thus this set of experimental data fit the Langmuir isotherm.
The second part of the experiment was conducted to determine the adsorption capacity of carbon by manipulating the amount of activated carbon in the solution from 300 gram to 500 gram while keeping the methylene blue concentration constant at 30 mg per liter. Activated carbon is a highly porous carbonaceous substance. Therefore, when more activated carbon is added, it provides a larger surface area for adsorption. As time increases, more methylene blue is being adsorbed, hence leaving behind a less concentrated methylene blue solution. To study the adsorption isotherm, we plotted the Freundlich and Langmuir graph. As you can see, the Freundlich graph does not obey the law, whereas the Langmuir graph gives the best fit for this part of the experiment with a regression value of 1.
Hi, as a conclusion, from part A, the manipulated variable is the initial concentration of methylene blue, while the constant variable is the mass of activated carbon. After plotting the adsorption isotherm, we found out that both describes the system well. However, we chose the Langmuir isotherm as the regression value is closer to 1. While in section B, in part B, the manipulated variable is the other one. It is the concentration, it is the mass of activated carbon, while the constant variable is the initial concentration of methylene blue. In this part, we also chose the Langmuir isotherm because the Freundlich isotherm does not comply to the theoretical value. Thus, from both experiments, we deduce that the best isotherm that fits our experimental data is the Langmuir isotherm.